Hello and welcome to the new topic, genetic diversity. As you know, we are studying nowadays different. We are studying the biodiversity and different types of biodiversity. So in our previous lecture, we studied the biodiversity, and the first type of biodiversity is the species diversity. We already studied the species diversity, how to measure the species diversity, and how to calculate difference between different ecosystem, measure the species abundance and species richness, and then scales of biodiversity, that alpha diversity, beta diversity, and gamma diversity, and the benefits of species diversity or benefits of the biodiversity. Today in our second lecture we will study the genetic diversity. As we already studied, the genetic diversity is the variation in genetic composition. Okay, it is the uh, genetic diversity variation in the genetic composition among individuals of a population or of a species. Okay, you can see the genetic diversity in the pictures given or in the example, that the differences between the species of giraffe. So in their color difference, in their pattern difference, so you can clearly see the difference in this within this population or within this species. So genetic diversity is the diversity or variation in the genetic composition among individuals, and it is also said that total number of genetic characteristics in the genetic makeup of species. So any diversity is the total number of genetic characteristics. Okay, the total number of genetic characteristics, the variation in the total number of genetic characteristics in the genetic makeup of a species or of a population. And we will study the genetic pool. A large genetic pool indicates that high genetic diversity increase chances of biological fitness and survival. This means that if there is the genetic variation are larger, if we have large genetic pool, the number of individuals are large, there is more genetic variation, then their fitness and survival is in the fitness and chances of survival are increased as there is more genetic variation among that population. Genetic pool, as we already know, that it is the alternative form of genes. A gene pool is collection of all genes and all the alternative forms of genes. The alternative form of gene is allele. As we know that in within uh, any gene there are the two different forms, or there are two alleles in that genes. One comes from the both of them comes from the parents, one from mother and one from the father. So these two alleles which comes from mother and father, these are different forms of alternative forms of genes, or these are the alleles. One can be both can be homozygous, both can be heterozygous. So a genetic pool is collection of all genes and all the alternative forms of collection of all genes or all the alleles in any population, usually a particular species. Then this can be in any population. For example, over here we have given the example of frogs in the ponds, trees in the forest, or people in the town. So they have different genes. So the collection of all those genes is the gene pool. Over here I have given the example. You have a population of cows of different colors. The colors are caused by different variation of the same gene. All the different variation of genes make up the gene pool. So all the variation within a population is the collection of all the genetic makeup, or collection of all genes within a population is the gene pool. So the gene pool does not bother with the frequencies. A variant, a gene is a variation can be present or can be absent. So it does not bother with the frequencies, its presence or absence. a variant can be present or it can be absent. So how much it is present, the gene pool doesn't count this thing. It is present or it is not present. The composition of a population gene pool can change over time through evolution. So evolution through natural selection or through different breeding techniques, the different environmental factors, the evolution can occur. So the composition of population's gene pool can change over time. Okay. So this means that the gene pool of a population can change over time. The result is a gene pool that is altered to be attuned to the needs of population specific environment. So in different environments, okay, it can be varied. Okay. The genetic variation, those are mostly caused by mutations. As you know that different and mutation occur due to environmental factors, due to different factors. For example, over here I have given the example migration of human population from equatorial region towards northern climates, where they were exposed to relatively low amounts of sunlight, resulting in change over time in the skin pigmentation, with skin becoming more lighter in color to augment vitamin D absorption. The genetic modification underlying the change in the pigmentation ultimately became a part of many of other population in gene pool. So I have given the example. In this example, you can see that the people who from the equator to the northern climates, when they migrated, their pigmentation or their skin pigmentation, they became lighter, and this is one of the example in the alteration of the gene pool. So now coming to the main topic, causes of genetic variation. Causes of genetic variation. Genetic variation are generally referred to the variation in genes between different individuals of a population. So variation in genes between different individuals of a population. Genetic variation is important for survival. Now over here I have given the example of evolution, macroevolution and microevolution. Microevolution within a species or within a population, and macroevolution from the start, from bacteria to fish, from single cell to double cell to triple cell, and then multiple cells organism, and then evolution of the human being. Okay. Genetic variation is important for survival and adaptation of species, and it helps in terms of natural selection and evolution. So we studied earlier that it is important, the genetic variation, according to the climatic changes, according to the environment, it is the genetic variation is important. Genetic variation is important as these genetic variation will increase the chances of survival and fitness, and these are caused by natural selection and evolution. Genetic variation are caused by mutation. Okay, are caused by mutation, genetic drift, gene flow, non-random mating, and natural selection. These are the causes of genetic variation. So the main cause of genetic variation is the mutation. The main cause in genetic variation is the mutation. So mutation is the main cause by which the genetic variation occur. Now we will study in detail all these: genetic drift, gene flow, non-random mating, and natural selection in detail. So let's have a look at the mutation. Mutation, there are two types of mutation. Mutation is the change in the genetic makeup, or variation in the change occurred in the genes or in the whole genome is the mutation. So gene mutation, we have two types of mutation, is the gene mutation and chromosomal mutation. Gene mutation are the small changes affecting the nucleotide sequence of one gene. Before going into the detail, you should know that the central dogma that during the process of transcription, translation, and then the synthesis of protein, that nucleotides, the nucleotide, they sequence for the production of mRNA, and then these mRNA, they cause the protein synthesis. Okay, then they cause the protein synthesis. The proteins are produced. So small changes affecting the nucleotide sequence of one gene is the gene mutation. The two main types of gene mutation are point mutation and frame shift mutation. Point mutation and frame shift mutation. We will study these point mutation and frame shift mutation in detail in the next slide. The next one is chromosomal mutation, or substantial alteration of the genetic material of an organism. Okay, some substantial alteration of the genetic material of an organism is the chromosomal mutation. This means that there can be the structural changes in the chromosome, or there can be the change in the number of chromosomes. Okay, the change in the number of chromosome or structural changes in the chromosome is the chromosomal mutation. Different causes, type of chromosomal mutation, or deletion of the chromosome, or duplication of the chromosome, and insertion, inversion, or translocation of the chromosomes. So this is the chromosomal mutation. Now as the gene mutation, these are there are two types of gene mutation: point mutation and frame shift mutation. So if the first one is the gene mutation can be caused by the insertion. What is insertion? For example, we have a codon A, C, C. Okay, it is this. If another codon is if another nucleotide is placed over here, A, C, for example, this is inserted inside A, C, T, C. So this is the T nucleotide which is inserted over here. It will cause the change in the sequence of the nucleotide. It will cause change in the sequence of nucleotide. So this is insertion. Deletion is the removal of single or very few nucleotides. For example, if I in the same example, if I remove the C and place it with the T, then and place it with the T, then it will produce. So for over here, if I remove this C, then it will produce A, T, C, this new nucleotide. The nucleotide is deleted. Substitution is the replacement. If this C is replaced by T, then this is substitution mutation. These changes can lead to point mutation. Point mutation can be silent mutation, missense mutation, or nonsense mutation. And insertion and deletion. Okay, insertion and deletion. This can be caused by the these insertion and deletion can also cause the frame shift mutation. It changes the whole frame, the whole sequence of the the whole sequence in the addition or at the end. It changes the whole sequence of the nucleotides, and it will produce a new type of protein. So let's come to the silent mutation. Silent mutation do not have any effect on the cell metabolism. Okay, so if there is coding is taking place, if there is any alteration in the nucleotide sequence, that nucleotide sequence, as you know that the codons, that if there is any change in the codons or in the nucleotide sequence, that doesn't lead to the change in the amino acid, that doesn't lead to the change in the protein synthesis, the specific type of protein synthesis, then this type of mutation is silent mutation. Okay, if there is no change in the protein synthesis, then this type of mutation is silent mutation, that it will not change the protein. Missense mutation, on the other hand, code for different amino acids. The result of these amino acids, that it will change the mRNA sequence, and the change in mRNA sequence, if it doesn't lead to the any alteration in the protein, but still it produced functional protein, then we can call this as missense mutation. Nonsense mutation, if it produced non-functional shortened protein. Okay, if it produced non-functional shortened protein, then this type of mutation is nonsense mutation. Nonsense mutation code for premature stop codons. Okay, premature stop codons, which will stop premature stop codon, which will lead to producing of a protein, shortened protein or non-functional proteins. So this is three types of point mutation. Then we have frame shift mutation. Frame shift mutation, as we already studied, that it is caused by insertion or deletion in the genome, or insertion or deletion of the nucleotides that are not in multiples of 3 nucleotides, that are not in multiple of 3 nucleotides. As a result of these insertion and deletion, the reading of frame of mRNA is changed, which result in protein with different amino acid sequence. Okay, so in frame shift mutation, a new type of protein is synthesized. It can occur at the start of the gene. If it occurs at the start of protein, then totally new protein is produced, or at the end of gene, if it is at the end of the gene, so it will change the sequence of only few amino acid. Okay, so a protein which have a different function, it will produce a protein which have a different function as compared to the original protein. So this is the frame shift mutation. Then we have chromosomal mutation. Chromosomal mutation, it can be deletion, inversion, insertion, or translocation. Okay, and duplication or amplification. Translocation, and the fourth one is duplication. So deletion is what is deletion? Deletion is the removal of some chromosomal region. You can see over here, I have given the examples, so you can study these examples of deletion, inversion, insertion, and translocation. You already know these things. So removal of large chromosomal region, leading to the loss of genes within that region, is the deletion. Inversion, reversing the orientation of a chromosomal segment. Okay, reversing the orientation of chromosomal segment over here. Inversion, this one is inversion. This segment, this is, you can see over here that this is the reversed, the sequence is reversed, so this is the reversing the orientation of the chromosomal segment. Insertion is the addition of material from one chromosome to another non-homologous chromosome. Insertion, this is from here, it is inserted into non-homologous chromosome. Translocation, interchange of genetic material between non-homologous chromosome. Okay, translocation, insertion of genetic material between non-homologous chromosome. So both the chromosome, they what they do, they interchange the genetic material, not the insertion, interchange of the genetic material between two homologous chromosomes is the translocation. Duplication lead to multiple copies of chromosomal region, increasing the number of genes located within that region. And there are different causes of gene duplication. Unequal crossing over between 2 and 3. You can see that between chromosome 2 and chromosome 3 there is unequal crossing over between 2 and 3, and you can see the result over here. Okay, mistakes during the this all occur, the duplication or amplification, this all occur during the meiosis, during the process of meiosis. Mistakes in DNA replication or DNA repair mechanism, and transposition. If, as you know, the central digma, it will produce the cDNA, and cDNA will go to the target site, and it will cause the transposition. It will be transported to the target cell, or whole genome duplication. Our whole genome is duplicated during this process. Hopefully you enjoyed the lecture. Thank you very much. In next lecture we will study the other causes of the gene variation. Thank you very much.